Good evening, everyone. Today we'll be reading a research paper, or not a research paper. This is whatever this is. This is a paper on Introduction to Chronobiology by Sandra J. Kuhlman, L. Michael Craig, and Jean F. Duffy. As a consequence of Earth's rotation about its axis approximately every 24 hours, most organisms on this planet are subjected to predictable fluctuation of light and temperature. A diverse range of species from cyanobacteria to humans evolved endogenous biological clocks that allow the anticipation of these variations. Thus, the internal physiology and function is fundamentally intertwined with this geophysical cycle. In fact, it was an astronomer, Jean-Jacques d'Ortus de Marienne, rather than a biologist, who provided early insight to this evolutionary relationship between internal physiology and geophysical cycle. De Marienne, in 1929, made the observation that daily leaf movement in heliotrope plants continue in constant darkness. To emphasize the endogenous and self-sustained nature of biological clock, Franz Hellberg in 1959 coined the term circadian. Latin. Circa means about and dies mean day, to refer to daily rhythms that are truly endogenously generated, that is, rhythms having a period of about 24 hours that continue to oscillate in the absence of any environmental input. Chandrasekharan, 1998. Rhythm generation is now understood to be an intrinsic property of single cells, driven by an intracellular molecular oscillator based on the transcription or post-translational negative feedback loop. Under normal conditions, these endogenous oscillations are synchronized to the environment, and it is generally thought that biological clocks provide an adaptive advantage by ensuring that an organism's internal biological and physiological processes, in addition to behavior, are optimally adapted to the local environment. So we'll see the historical timeline now. As early as 1729, it was documented that this telirhythmic behavior was likely endogenously generated, and it was also near this time that Carl von Linné, 1707 to 1778, constructed a floral clock, noting the predictability of petal opening and closing times of various species of flowers. Chandrasekharan, 1998. However, it was not until 200 years later that Erwin Bunning provided the first evidence of the genetic basis of circadian rhythms generation by demonstrating that period length by heritable that period length is heritable in bean plants. Bunning also put forth an influential hypothesis that circadian oscillators can be used to measure seasonal changes in addition to measuring daily cycles, and pointed out the adaptive significance. What is happening? Wait a second. The adaptive significance of tracking seasonal changes. Okay. Thus, the field circadian rhythms originated from keen observation of plants, and it was not until later that the first observations of endogenously driven rhythms in bacteria, Mitsu et al. 1986, single-cell eukaryotes, Sweeney and Hastings, 1957, insects, Belling, 1929, birds, Kramer, 1952, rodents, Richter, 1922, primates, Simpson and Gaybrand, 1906, humans, Escoff and Weaver, 1952, was discovered. A breakthrough in understanding the genetic basis of rhythm generation was made by Ronald Konopka and Seymour Benzer. Understanding the genetic. Using a mutant screening in Drosophila melanogaster, Konopka and Benzer, 1971. Mutagenized flies were examined for the persistence of two circadian behavior, pupil exclusion, pupil eclosion and locomotor activity. Flies displayed one of three categorical mutant phenotypes. Lengthening of circadian period, shortening of period, and erythema. All phenotypes were complemented by a single locus, now referred to as the period gene. Shortly after this discovery in fruit flies, the frequency gene was shown to be essential for rhythms in conidiation to persist in the filamentous fungus, Neurospora crassa, Fedel and Hoyle. These results showed that single gene mutation could disrupt a complex behavior. Together with the discovery of heritable timing mutations in hamster by Martin Ralph and Michael Menker. (1988) provided the rationale for pursuing large-scale mutant screening in mice. Subsequently, using a variety of strategies, dozens of clock genes have been discovered in both prokaryotes and eukaryotic system, including cyanobacteria, fungi, plants, insects, and mammals. Even human rhythms are potentially altered by a clock gene mutation. Jones et al. 1999. A striking common principle emerges from the examining these diverse clock gene systems, that all organisms seems to have evolved transcriptional or post-translational feedback loop. Where is the marker? Where is the marker? Okay. Principle emerges from the examining these diverse clock gene system is that all organisms seems to have evolved transcriptional or post-transcriptional feedback loops to ensure high amplitude near 24-hour rhythm generation. Furthermore, the identification of specific clock gene has led to the development of real-time bioluminescent and fluorescent reporters that have spatial resolution to track rhythmicity at level of functional unit of rhythms generation, that is a single cell. Hastings et al. 2005. Such resolution is essential to understand how individual oscillators are coupled with population of rhythmic cycle cells. However, in the midst of this cycle gene explosion, there is an important lesson to be learned from a pivotal experiment conducted using clock components of cyanobacteria. In 2005, Nikazima and colleagues constituted a circadian oscillator in a test tube using only cyanobacterial proteins and ATP. The lesson: it is possible to construct a near 24-hour oscillator in the absence of gene transcription. Indeed, circadian research has expanded in recent years to include a system level perspective on the independencies of cellular metabolic networks such as rhythmic redox cycles and transcription translation feedback loops. It is clear from the work of many talented and dedicated scientists that most organisms inherit the innate ability to keep track of time on 24-hour scale. How do organisms use their timing devices? What are the negative consequences of organisms if organisms are no longer able to effectively use their clocks, such as in diseases or aging? We know from the work of Carl von Frisch and Ingeborg Belling that bees use their clocks to visit flowers at the appropriate time of the day so that they can feed when flower is open. Work from Gustav Kramer shows that birds use their biological clock during migration to help compensate for the changing position of the sun throughout the year. Additionally, de Courcy showed that chipmunks use an innate biological clock to properly time foraging so as to avoid predation. And studies on monarch butterflies reveal that the circadian cycle participates in initiating migration by tracking seasonal changes. Future studies in the field of circadian rhythms will continue to explore the way in which organisms use their biological clock to coordinate internal functions, time their behavior, and navigate from the environment. So let us see this diagram. Figure 1. So what is it saying is transcription translation regulatory feedback loop. This is a transcription translation regulatory feedback loop, a common theme in the generation of eukaryotic circadian rhythms. Many circadian systems are composed of positive and negative elements that are involved in the feedback loops. Positive elements oval interact and bind to the upstream elements white rectangle oval and upstream elements white rectangle may be these. In the promoter region, where is the promoter region? I don't know. Bent arrows, this or this. Of gene that encodes negative element to activate their expression. These negative components tetrahedron interact to inhibit the activity of positive elements. They inhibit the activity of positive elements. Okay, wait a second. Once more. So positive elements oval. These are the positive elements. Let me clear this out first. So positive elements ovals. This is an oval. Positive elements. Positive elements interact and bind to upstream elements white rectangles. So they coming here and they interact with the upstream elements, the white rectangles. They are interacting with them. Okay. White rectangles in promoter region. So I'll say this is the promoter region. What next? Bent arrows. They are saying promoter region is bent arrows. I do not understand. Okay, this is the promoter region. Okay. I was wrong here. So positive elements interact and bind in upstream elements, rectangles, in the promoter region of genes that encode negative elements. So they encode now negative elements to activate their expression. Okay. These negative components, tetrahedron. So the positive elements came and interacted with these upstream elements. They went to the promoter regions and they encoded the negative elements. Now these negative elements are going interact to inhibit the activity of positive elements. Now they are interacting with each other. Now they will inhibit the activity of positive elements, these positive elements, which ultimately leads to decrease in expression of negative elements. So now they are here, they are affecting, they are stopping the positive elements. So this will reduce making of the negative elements, so which will decrease interaction of negative elements. I think they are saying that decrease in expression of negative elements. Progressive phosphorylation P of negative elements lead to degradation and relief of the inhibition of positive effectors. So some went here to affect our positive elements, but some are phosphorylation induced degradation. So P comes here and P just degrades them. Progressive phosphorylation occurs of negative elements that leads to their degradation. They are degraded here. So this is a relief for the positive effectors to allow the cycles to start once again. Okay, that's nice, that's cool. Other components, diamond. This is diamond and this is diamond. Where is it? May be present that bind to the promoter of genes that encode the positive elements to comprise an additional feedback loop. Okay, so they were already coming here to the white rectangles, the upstream elements. Let's call them upstream elements, which were then going for the this is the promoter region. And then these diamonds that are present, other component, they are sent back, they form an additional feedback loop. Okay, they may bind to promote genes that encodes, they may bind to promote genes that encode the positive elements. So now the formation, this is the formation of positive elements. This is additional feedback loop, this was negative feedback loop. Because of this, the this was AB part was decreasing, and because of this the AB is forming. So this is what it is. La 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 la. Okay, understood. Nice. The arrows and perpendicular lines indicate positive and negative regulation. respectively. Okay, next. Here, circadian terminology. Here we define some commonly used terms in the field. Black box. Experimental designs are frequently used to probe the mechanisms underlying clock function. For example, de Marian used a black box approach in his heliotrope plant study. The plant was treated as a system whose internal components were unknown, that is black box, but whose function was studied by assaying the observable output of leaf movement in response to perturbation caused by environmental inputs, the light and dark cycle. To a large extent, the following terminology developed over the years to precisely report the results of black box experiments. Observable or measurable output rhythms of the circadian timing system, such as leaf movements, are defined as overt outputs. In the case of animals, wheel running activity and levels of circulating hormones in the blood are two commonly assayed overt outputs. In addition, a system can be composed of multiple oscillators in which the output signal of one oscillator influences the circadian property of another oscillator. The output signal can be referred to as coupling signal. In practice, the distinction between an overt rhythm and coupling signal may depend on experimenter's perspective. A rhythm is considered to be circadian if the oscillation has a period approximately 24 hours and continues in constant conditions, such as constant light, referred to as LL, and constant dark, referred to as DD. The inability of a rhythm to persist under constant conditions implies that the rhythm is driven by responses of periodic external time cues or Zitterberg. Or periodic changes in behavior rather than generated by periodic internal process. It is important to keep in mind that when rhythmicity is measured at a tissue or population level, the loss of an overt rhythm owing to experimental perturbation may not be the result of loss of rhythmicity per se. Rather, individual oscillations may continue but not observed because of phase difference among oscillating units. The time needed for one cycle of a circadian oscillator to occur in constant conditions is known as free running period. The time needed for one cycle of circadian oscillation to occur in constant condition is known as free running period. Under normal conditions, circadian rhythms are not free running, rather they are synchronized to the local environment. The process by which a rhythm synchronized to an external cycle is referred to as entrainment. Interestingly, although circadian rhythms persist in constant conditions, the FRP can vary slightly in response to changing light intensities. Diurnal organisms display a slightly shorter FRP, that is faster clock, in higher light intensities than they do in low light. Nocturnal organisms show inverse response with the longer FRP, that is slower clock. In highlight than in dim light. This phenomenon originally described by Jorgen Ashkoff is known as Ashkoff's rule. Ashkoff and Weaver in 1962. FRP can also vary based on the light history that the organisms experience before being placed in constant conditions. The influence of light history on FRP is referred to as an after effect. Clearly, when assaying overt rhythms and calculating the FRP, the influence of these factors should be taken into consideration. And in addition to being endogenously generated, there are two other properties of circadian rhythm that are often focused on experimental studies. The ability of the rhythm to be reset or phase shifted by transient exposure to complete to time cues such as light, and temperature compensation. Once more, in addition to being endogenously generated, there are two other properties of circadian rhythms that are the focus of experimental studies: the ability of rhythms to be reset or phase shifted by transient exposure to time cues such as light, and temperature compensation, defined below. Phase shifting and phase response curve, PRC. The phase of a rhythm can be shifted or reset by transient exposure of certain environmental cues. Because FRPs are close to but not exactly 24-hour, the rhythm must be reset each day to avoid falling out of sync with the external environmental cycle. As discussed earlier, many organisms use the daily alteration of light and darkness as the environmental signal to entrain their rhythms to a 24-hour day. An early study by Hastings and Sweeney, 1958, in the dinoflagellate Goniolax polyhedra showed the property common among circadian systems. Their biological clock is not equally sensitive to light at all times of day. As has been shown subsequently for many organisms that use light as entraining signal, light exposure in the early subjective night produces a phase delay shift, that is, rhythm is delayed to a later hour, whereas light exposure in the late subjective night periods of phase advance shift. The rhythm is advanced to an earlier hour, and light during the subjective period produces very small to no phase difference. Thus, the response to light is dependent on the organism's internal sense of time. The multitude and direction of these responses can be plotted with respect to organism's own circadian phase at which the light was presented, and such a plot is referred to as a PRC, phase response curve. So let us see, here are few phase response curves. Properties of circadian rhythm. A. Behavioral circadian rhythm can be entrained by external stimuli. Such a light-dark cycle will persist with a near 24-hour period in the absence of environmental cues, such as constant darkness. Properties of rhythm that are commonly measured are period, phase, and amplitude. Period is the duration of time to complete one cycle. It is typically measured from peak to peak. Let us see. Peak to peak, a period. a period. So from this peak to this peak, this is a period. From this peak to this peak, a period. But it can be measured from any specific point on the curve. So we can also like take from here this point to this point, because after this it will be repeating the cycle here, then repeat and then repeat. Okay, so this is a period. Phase is a relative position on the curve. This is a relative position. This is a phase shift. Example, the peak in reference to a particular time, such as time placed in constant darkness. Amplitude is the measurement of recorded output from midline of the curve to either peaks through or trough. Amplitude is a measurement of recorded output from the midline. This is amplitude, midline, the row and trough, the peak and trough. B. So this is what we were seeing in A. So this is periodic. This whole thing is periodic. Why? Because there is a regularity in this cycle. We see enough peaks and troughs. There is a period. The maximum difference between the peak and the trough is an amplitude. This is a phase, like a light phase and or a dark phase. Okay, and if this started something like this, then it will be a phase shift. How much phase shift? This much phase shift. Okay, let's now read more. So in x-axis we have time, in y-axis we have clock controlled output. So B, the phase of a circadian rhythm can be reset by a stimuli to which it entrains. In this case, exposure to a stimulus input rapidly lowers the level of rhythmic variable dashed line, which recovers to a rhythm that with a shifted phase as compared with the curve that did not receive input. On x-axis, time in the light-dark cycle is depicted by alternating white or black bars. Time under constant conditions is depicted by alternating hatched and black bars. Level of clock controlled output is displayed by y-axis. See, so they are showing phase shift and the input. It is starting from here. This is the dark light part, this is the dark part. See, phase response curves measure the magnitude and direction of phase dependent responses on the brief exposure of external stimuli. This is the day part, this is the night part. Okay, there is delay, below this is a delay, and above this is advance. We are seeing the phase shift. So let us see the B once again. A normal cycle was going on. Okay, we put an input. There was an input which changed this flow to a different one. So now we see there is a phase shift. of this much. This is a phase response curve, C1. Once again, we are seeing that only. A phase response curve measures the magnitude and direction of phase-dependent response to brief exposure of the external stimulus. The x-axis represents the circadian time at which the light pulse is applied to the organism. Then y-axis shows the change in the phase of circadian controlled output in response to the light pulse. The positive shift are indicated as advances, positive shift advances, and negative phases shifts are delays. So we can say here it is a positive shift, there were advances, and now here is the negative shift. We can say there are delays, and then again some advances. A brief light pulse given to an organism during the subjective day, organism's own internal day produce little to no phase response. Light in the early subjective night, the organism's own internal light produces phase delays, and light in the early subjective night produces phase advance shift. Light in the early subjective night, light in the day, light in the late subjective night. Okay, so if an organism whose, let us say there is let us say the normal organisms go to sleep at 6. Okay, so if we are not giving them anything, they'll go like this. Okay, no problem to them. If they go to sleep at 5, okay, so it is what it is called this early subjective night. Sorry, this is late subjective night, and we give them light. Okay, it is complicated. Whatever, let me see. So if this person sleeps at 5 and we are still giving it light, so this will think that it will sleep late, so there is a delay, so we'll go down. Okay, and if this one who sleeps at 7, we are giving it light again at 7, it will be show some advancement. I don't know, it said something. Okay, next page, figure number 3, importance of assaying rhythms at single cell resolution. To be considered circadian, a rhythm must be maintained under constant conditions. Okay, there must be a rhythm. When examining rhythms at the tissue level, in which the tissue is composed of multiple cells, it is important to realize that the absence of rhythmicity under constant conditions could be attributed to two possibilities. One, either the loss of rhythmicity, indicating the rhythm is not circadian, or dysynchrony among oscillators. In the latter case, individual rhythms may actually be circadian. However, the assay has insufficient resolution to show the presence of rhythmicity. To distinguish between the two possibilities, assay sensitive to the functional unit of rhythm generation must be used. So this is average signal, individual unit signal, with signal loss of rhythmicity. They lost the rhythmicity. Okay, they lost the rhythmicity. Or dyssynchrony. It was supposed to be like this, but it is like this now. There's a dyssynchrony. It is important to realize that there are cases in which it is possible that exposure to an external cue will simply transiently perturb the level of the rhythm being essayed without leading to a persistent shift in the rhythm. This condition is referred to as masking. Mechanistically, masking can arise because sensory pathways may act on a specific behavior independent of the clock. Temperature compensation. For a biological clock to be reliable in a natural setting, its period should not change much despite change in the ambient temperature. Morning events should occur at essentially the same time, independent of weather and conditions the night before. This property termed temperature compensation stems from the observation that the value of the FRP changes very little during the exposure of different temperature within the organism physiological range. The effect of changes in temperature on the rate of the most biochemical reactions is measured by Q10 value, which is defined as the ratio of the rate of a given process at one temperature to the rate of temperature 10 degrees lower. The Q10 value of period of a circadian remains near 1, as opposed to other known biochemical reactions that have Q10 values of 2 or 3. However, temperature compensation is not the same as temperature insensitivity. Temperature itself can be strong Zytoberg in some organisms. Individual reactions within the clock are undoubtedly affected by changes in temperature, but the system as a whole is buffered such that the output of rhythmic behavior does not show large variations in its FRP. Concept of a clock. What is a circadian clock and how has the concept of clock facilitated research? As described in one account, Chandrasekhar in 1998, the now widespread use of the term clock was in part inspired by Kramer's studies on time compensation of the sun compass in the birds. Shortly thereafter, in 1960s, the Cold Spring Harbor Symposium of Quantitative Biology was boldly titled Biological Clocks. Today, the term clock is used pervasively to emphasize the endogenous nature of rhythm generation. That circadian rhythms are innate rather than learned or passive phenomena, and to imply the primary function of circadian rhythms is to measure time. It is this conceptual framework that prompted the design of shifted schedule or jet lag experiments and helped us understand the relationships between seasonal adaptation and circadian rhythm generation. Conceptually, the components of a circadian clock can be broken down into three basic elements: an input pathway, a pacemaker, and output pathway. At the heart of a circadian clock is a pacemaker, a central oscillator or a network of coupled oscillators that is entrainable and has the ability to synchronize downstream targets. Temporal information provided by pacemaker is interpreted by output pathways. which then regulate the timing of metabolic and behavioral processes. For the oscillator to maintain in sync with environment, input pathways must relay external timing cues to the pacemaker. Although conceptually, one can discuss these three elements as distinct entities in biological terms. One protein or physiological process can serve multiple roles. A central pacemaker can coordinate numerous peripheral clocks. Within multicellular organisms, individual cells possess the ability to oscillate on a circadian scale and are themselves bona fide clocks in the sense that their gene transcription translation feedback loops are entrainable by factors such as internally circulating hormones, and they are that are capable of producing rhythmic outputs in gene expression and protein function. Typically, the circadian oscillations that exist within an organism are hierarchical, arranged and regulated by discrete identifiable tissue or small group of cells that act as a central pacemaker, sometimes referred to as a master pacemaker, capable of maintaining synchrony among the various clocks. Evidence for a hierarchical organization of clock comes in part from inactivation and genetic manipulation experiments. In mammals, inactivation by lesion, for example, of the suprachiasmatic nucleus results in loss of rhythmic outputs, including the sleep-wake cycle, wheel running behavior, hormone production, core body temperature, and accumulation of clock-controlled messenger RNA and protein at the organ and tissue level. Once again, in mammals, inactivation of the suprachiasmatic nucleus results in loss of rhythmic outputs, including sleep-wake cycle, wheel running behavior, hormone production, core body temperature, and accumulation of clock control messenger mRNA and protein. Accumulation of clock controlled messenger mRNA and protein at the organ and tissue level. Okay. Similarly, in Drosophila, genetic ablation experiments established that the ventral lateral neurons LNVs are essential for self-sustained behavioral rhythms. Okay. In addition to receiving the timing information of central pacemaker, peripheral clocks are often positioned to receive timing information from other periodic signals such as luminescence level, food intake, and locomotor activity. Thus, although central pacemaker exists, they rarely have exclusive control over many clocks found within the body of a multicellular organism. Furthermore, the coupling strength and more oscillators of the central pacemaker as well as between the central pacemaker and downstream targets can vary, and this variation may promote adaptation to changing environment such as day length. So there was a diagram we missed in the previous page. So this represents this is the representation of circadian clock divisions. A circadian clock can be depicted as having input pathways, a central oscillator, there is input pathway, central oscillator or pacemaker, and output pathways. The central oscillator produces the endogenous biological rhythms and can be synchronized with the environment via input pathways through cues such as light or temperature. Okay, so the cues are light and temperature, and it will tell the central oscillators which conveys it pathway, output pathway. Okay, output pathways convey clock's rhythm to downstream targets. Okay, it will tell clock controlled activities targets and derive overtly rhythmic activities. Some circadian systems consist of more elaborate pathways shown in the dashed. This is a more elaborate pathway that include multiple interlocking oscillators and positive negative feedback from clock controlled activities to oscillator. You can go here also, or input path components. Okay, so what is happening here? They are telling us there is input pathway, there is a central oscillator or a pacemaker, and output pathways. The light and temperature comes through input pathways. They are signals sent to the central oscillators, and then output pathways are told, which tell the clock control, which puts out the clock control activities, which convey the clock's rhythm to downstream targets and drive overtly rhythmic activities. Feedback and rhythmic transcription of input pathways. They go through here, or they take a long route via many interlocking circulators, gives feedback, creating back to input pathways. I do not understand why they are doing this, but they are doing this. Okay, another circadian timing in humans. Human living in industrialized society with widespread use of artificial light around the clock jobs and social activities and ability to travel rapidly across time zones are often exposed to conditions in which the internal circadian rhythms become desynchronized from the experienced light dark cycle. There is now increasing evidence that disruption of internal rhythmicity have a negative health outcome in people, including insufficient sleep and corresponding drowsiness, associated safety issue, increased susceptibility to immune challenges, increased risk of cardiovascular and metabolic diseases, and increased cancer risk. These disorders are thought to arise from disruption between central CNS pacemaker. What is the CNS? Suprachiasmatic nucleus, and peripheral clocks, attributed to mismatch between environmental light dark signals behavior and internal oscillators. There is also evidence that dysynchronization of individual cells within the organ can occur, leading to inefficient function of that organ. As our methods of assessing circadian rhythmicity in humans in the field and clinic improve, the field of chronomedicine will be able to develop, as will chronotherapies to treat circadian dysynchrony. Applying our understanding of circadian timing system to the environment of human disease holds huge potential for improving health through personalized medicine. The potential for such therapeutic advances in is highlighted by recent study that found substantial overlap between gene and that are rhythmically expressed genes that are associated with disease and genes that are targeted for best-selling medications. More than half the best-selling medication in United States targets the genes that oscillate in the circadian manner, and more than 100 of the World Health Organization's essential medications target circadian genes, indicating that the pharmacokinetics and pharmacodynamics of those medicines are likely to vary with time of day. However, clinical and preclinical drug studies do not typically vary or attempt to optimize the time of day at which medications are administered, leaving open to the possibility that efficacy could be improved, side effects could be reduced, or both if the ideal time of day for administration were known. Moreover, with the development of methods for rapidly and easily assessing circadian timing in the field of clinic and timing of medication administration will be able to personalize account for individual differences in circadian timing. Conclusion: in summary, circadian rhythm research has revealed how cellular processes interact to produce daily rhythms and physiology and behavior that can increase witness across diverse range of organisms and species. 300 years on, we are now at cusp of harnessing this knowledge to reveal fundamental properties of cellular network from a systems biology perspective to improve human health. This figure 5 schematic representation of the overlap between circadian gene and those with important medical relevance. This figure shows the overlap between genes that have a circadian oscillation, red, those that are associated with diseases, tan, and those are targets of 100 top selling drugs in the United States, blue. Few of the drugs are prescribed with specific timing information representing potential targets for development of chronopharmacology to improve efficacy and or reduce side effects. So white is all genes, red is circadian genes, blue is target genes, drug targets, and this green or gray or what is this cream color, tan color is genes associated with disease. So with this we are done with this paper. Thank you. Have a brilliant day. Bye bye.